Hello and welcome to the video abstract for our paper titled Loss of the NF-kappa B negative regulated PERK in Drosophila links brain and gut immunity to neurodegeneration. Today I'll be talking on behalf of the Ligoxygarchus Drosophila Immunology Lab at the University of Oxford. Prior research indicates the role of a dysregulated immune system in connection to age-dependent neurodegeneration, with recent evidence of transcriptional changes in immune signaling pathways in patients with late-onset Alzheimer's. Our research focuses on the use of the innate immune system in Drosophila to explore if this is an evolutionary conserved pathogenic route of neurodegeneration. I shall start by explaining why Drosophila is such a great model organism for our research. Genetic modification is simple. They have a small, fully annotated genome and a powerful genetics toolbox for gene modification. They also have a short generation time of 10 days, high fecundity, and ability to produce progeny from single-fly crosses. Their short lifespan of up to 50 days is also ideal when studying age-dependent effects. Their glia share many characteristics with human microglia, immune cells which have been highly implicated in Alzheimer's disease, and it is important to note that they do not develop amyloid plaques or tau protein tangles, which are common characteristics driving the protein aggregation hypothesis. Because of this, we are able to explore other factors that contribute to a neurological phenotype independent of protein aggregation. Drosophila lack adaptive immune responses, but their innate immune system shares many evolutionary conserved components with humans, as demonstrated here by their homologous tumor necrosis factor receptor 1, or TNFR1 pathway, named the immune deficiency or IMD pathway in Drosophila. TNFR1 operates in many organ systems, and predominantly in the immune system. The IMD pathway is shown in more detail here. To prevent an overreactive immune response to commensal bacteria, this pathway is highly regulated, with some of the key intracellular negative regulators shown in red. There is evidence to suggest that Drosophila mutants of immune genes have corresponding neurological phenotypes. Many of these immune genes are in fact negative regulators of the IMD pathway, also highlighted in red, and there was additional evidence that these phenotypes were recoverable in axenic conditions only with PERK. Hence, manipulation of PERK was selected as the means for generation of an overreactive innate immune response and to enable exploration of the potential role of microbiota. The gut-brain axis is an important concept that capitulates the potential interplay between the microbiome and brain activity. Loss of gut wall integrity, for example by inflammation, promotes movement of bacterial-derived products out of the gut, where they can then induce changes in the central nervous system. Mutants we used include a transposon insertion into the PERK gene to generate a PERK knockout mutant. We also used an imprecise excision of this transposon insertion, which demonstrated a dose-dependent neurological phenotype that correlated with PERK mRNA levels. And we used RNAi knockdowns, which enabled us to explore contributions of both gut and brain-specific cells. We did this by combining transcription factor Gal4 lines specific to intestinal stem cells, glia, and neurons with an upstream activating sequence targeting the PERK gene. We measured age-dependent neurodegeneration in the following ways: via behavioral assays, quantification of lesion formation in the brain, observation of microbiota dysregulation, and also by the ability to recover the neurological phenotype through axenic conditions and knockout of the antimicrobial peptide gene AttacinD, which is a downstream product of the IMD pathway expressed in the fly brain in an age-dependent manner. We carried out experiments at day 6 and day 28, representing early and late time points. I will now present some of our key findings. We demonstrated age-dependent decline in locomotion, in addition to a reduction in locomotion due to PERK knockout and knockdown. We also showed dysregulation of sleep. PERK mutants exhibited more sleep over the day at both early and late time points. Sleep is an interesting factor that is heavily intertwined with immunity and that poses a possible role as a biomarker of neurodegeneration. Further to this, we found increased number of brain lesions in PERK knockouts compared to wild type. And with regards to cell-specific knockdown, we show evidence for earlier onset of the neurological phenotype with PERK knockdown in intestinal stem cells compared to glia, as shown in their locomotion. Meanwhile, there appeared to be no effect at either time point when PERK was knocked down in neurons. As for sleep, there were increases in daytime sleep when PERK was knocked down in intestinal stem cells and glia, matching data for complete PERK knockout, whereas our PERK knockdown in neurons did not match this pattern. Evidence for the microbiome showed an increase in density and decrease in diversity in the guts of PERK knockouts at a later but not earlier time point. Acetobacteriaceae, a bacteria previously associated with aging and shown in brown here, dominated the microbiota of PERK knockout mutants. Axenic conditions also led to a partial rescue of the neurological phenotype, as shown here by locomotion data. Meanwhile, deletion of the antimicrobial peptide gene AttacinD recovered locomotion at a later time point, in addition to fully recovering lifespan, restoring daytime sleep patterns, but not nighttime sleep, and recovering brain lesions. To conclude, our results provide evidence for an evolutionary conserved path to neurodegeneration linked to dysregulated immunity in the absence of beta-amyloid or tau aggregation. We also show the role of the antimicrobial peptide AttacinD and the microbiome as potential mediators of this process.